Do it, computer. You got this. Here we go. Okay. Okay, so thank you guys for being so wonderfully patient while we cleared that room of physics or whatever we were doing. So in case you don't know, I'm Carl, one of the undergrads.
I'm Samira.
So we are going to work through the review tonight. So just a couple things. I do this every semester. It'll work. So you don't have to read all that. It's just my warnings. As undergrad TAs, we do sit in class. We go through the material with you guys. But we are not the final authority. What that basically means is what we say should be right, but if something doesn't really get clarified correctly, it's not really on us. So what that means is essentially if you are taking the exam and we say the sky is green, and you put that on the exam, if you get it wrong, you can't go back and say, oh, well, the TA said this, so I have to get points back. We're here just to kind of convey the information to you. If something that we're saying sounds wrong, like you know that the sky is blue and we're saying it's green, point that out and we will make sure that we're saying it right. We do this a lot, so we know what we're doing, but just don't rely 100% on what we're saying. So I'm like totally down talking us right now. Basically, we know what we're doing, but if we're explaining something that's confusing you, go back to Laura, talk to her, go back to your notes, use the lecture notes, things like that. Don't just take it at face value. If something doesn't sound right, tell us. In addition, I get this every semester about these PowerPoints. I'm not going to send these out. So what I did to make this was I went through the slides one by one and just pulled information verbatim from the slides. So I don't send this out because you guys can do that too. So just jot down the important stuff. You don't have to take down every single thing that I say, that we say, that's written on there. It's all on your slides. So just kind of listen, make sure you're understanding, and write down what you think is important. Any questions before we start? Yes? Is it going to be what?
Like a lot different.
Yeah, so the way she runs hers is she just does Q&A. And we've done Q&A before, and it just kind of gets chaotic, so I made a review session. Okay, it's not working. Hold on. Okay, well that's supposed to pop up. But basically it's just all the topics that we've covered so far, and we're just going to go through with all the information there, and you can ask questions as we go. So it kind of gives you a chance to ask questions, you get to see the information in quick relay, and we can go from there. So let me just get this to work again. Bear with me. Okay, so basically these are all the topics that we've covered. We're going to talk about cells, stem cells, IPS, signal transduction, all that kind of stuff. So if we don't talk about something and we talked about it in class, let us know. This may not have everything on it, but it's just taken from the slides. Okay. So cells. Hopefully we're all good with this so far, but they're basically complex living things that can independently reproduce. She calls them bags of proteins, and they're used to carry out functions in the body. There's prokaryotic and eukaryotic cells. There's different types of cells, differentiated, stem cells, stuff like that. So stem cells. You guys want to talk about stem cells?
Stem cells basically they have the ability to divide and or differentiate they divide, while terminally differentiated cells are just like differentiated into like a skin cell and that's it. They can't differentiate any further. But stem cells, they divide or differentiate.
So keep in mind that with stem cells, these are the ones that when we talk about cell division, these are what's undergoing it. Your skin cells, like right here, they're not changing. They're not all of a sudden going to be like, hey, let's become a brain cell. Let's go back and start dividing. Once they get to that point, they've passed go. They've collected their $200. It's over. It's done.
So there are 3 different types of potencies, or like basically the different level of what a stem cell can divide into. So you start with totipotent, which is it's basically a stem cell that can become anything. It can become the fetus or the extra embryonic cells or the extra embryonic placenta, umbilical cord, all of that. So the totipotent cell can do anything. And then you have pluripotent cells, which are essentially the same, but the one key difference you want to keep in mind is that they cannot become extra embryonic cells. They can still become the fetus. And then from there, they can differentiate further into bone marrow or anything else.
Keep in mind that you can't have a fetus if you only have pluripotent cells. So you need to have totipotent cells for a fetus to grow because you cannot have the extra embryonic tissue made from the pluripotent cells.
Right. So not all the extra embryonic cells are going to go on and become pluripotent. A bunch of them will stay extra embryonic and become, totipotent will become extra embryonic. And then multipotent stem cells they're more classified. So you'll have a group of cells that can become bone marrow, a group of cells that can become brain cells. You're within groups for multipotent stem cells. So you can't have a bone marrow cell become a stem cell. So basically, the multipotent cells are as they're as classified as it gets, and you can't really have them go randomly.
So one way to visualize this is if this line is all the options that a cell can become, totipotent can be anything on this line, anything in the realm. The line gets smaller for pluripotent. It can still do almost everything, but you can't do the extra embryonic tissue, so it's a little bit smaller. And then it gets even more condensed for multipotent because it's, again, really limited. So if you think of like very expansive totipotent and it slowly just kind of breaks down. You can also think that like a pyramid, other shapes, whatever helps you, but just kind of think totipotent's on top, multipotent, there's less options you can do.
As for multipotent stem cells, can they become anything just like pluripotent stem cells?
Multipotent?
Yeah.
So within our realm, so they can't become necessarily anything. So the one example we talked about for multipotent was hematopoietic stem cells, which become the blood cells. But you can't, again, make a fetus from multipotent. You can't make certain tissues from that. It's like very distinct.
I know totipotent is only in like your body for like the first 5 days of when you're pregnant and stuff. So pluripotent and multipotent, are those always in your body?
No, it's only the multipotent that
They're always
in our adult human body, basically.
So the totipotent's at the start. Pluripotent is through development as you're developing, you're getting these tissues. And then when you're in your adult body, we have multipotent stem cells in our bone marrow and other places, but that's about it.
The exception to that is a pregnant woman has extra embryonic and pluripotent, but that's for the sake of the fetus, and once the baby is born, she no longer has those.
Okay, so we talked about DNA a little bit. Something that is a common misconception is that stem cells, all cells have the same DNA. So DNA is present everywhere. The exception is red blood cells. Red blood cells don't have DNA, but don't worry about that. So remember that cells express essentially different genes. So depending on the genes being expressed, it's how the cell is. It's different combinations give you different cells. So just keep in mind that your DNA is in all cells. It's not in some, it's the same everywhere. Everything has the same genome. And we'll get more into this for exam 2 in terms of if we all have the same DNA, how does that make differences? Okay, so there's some different types of stem cells. Embryonic stem cells, mostly inner cell mass of the blastocyst, and to obtain them, you need to destroy the embryo. That's the ethical concern. Those are usually totipotent stem cells, which is why everyone kind of wants to use them for all these stem cell therapies, but you can't just destroy an embryo for that. iPS cells, there's another slide for those, but those are differentiated cells that are being induced to become pluripotent again. And then adult stem cells, like bone marrow, those are multipotent, and they can be used in tissue repair such as the liver, spleen.
So iPS cells are induced pluripotent stem cells. So the idea with this is that you're taking adult cells out of the body and genetically reprogramming them into an embryonic stem cell so that you can reintroduce it to the body and it can go on and divide and differentiate the way that an embryonic stem cell would. So some of the reasons why this is an advantage for researchers to use is because, well, first of all, with embryonic stem cells, you're taking the cells out of the body, so you're killing a fetus, and that causes moral dilemmas. Another reason why iPS cells are highly looked towards is because you're basically taking out the cells from the body and you're reintroducing it to the same body, which means that the immune system isn't going to reject it. If you're taking an embryonic stem cell from another body, reprogramming it or putting it back in, there's a higher probability that the immune system is going to recognize it as foreign and attack it. However, these are more difficult to make. They're more difficult, they're more costly, and they're not 100% the exact same as an extra embryonic stem cell. Yeah. So they're not the exact same. So that's one of the kind of cons with the whole concept of IPS.
Yep.
How are they not true, like pluripotent? Like, not true.
So true pluripotent stem cells are the pluripotent stem cells, whereas these, as Samira was saying, are formerly differentiated cells that you're putting back to be. So I mean, you're changing all the right genes, but because they're modified in like a lab, they're not true pluripotent. They're not going to be. You're not necessarily going to get the same results because they're engineered that way.
Why is one of the cons that they could be older cells? Because remember their lifespan is different from embryonic stem cells. I can't remember shorter ones.
So the longer a cell is alive, essentially, the more mutations it can accrue. So if you take a cell that has just differentiated into a skin cell, make it an iPS cell, you're probably going to be more successful than if you take a really old skin cell that has, think of our skin, we are exposed to sunlight, which is something that causes mutation, or basically, the older your cells are, the more mutations they can have, and that can be costly because mutations can lead to cancer, and then if you have the fact that it's now a pluripotent stem cell, it's just kind of not a good situation. So older cells could be a problem.
So can you turn any adult cell in your body into an iPS cell?
In theory.
Okay.
I know that they've done skin cells. I don't know if they've done heart cells, liver cells, stuff like that.
So most of the time it would be just skin cells into iPS cells?
I mean, I feel like that would probably be the easiest because if you do any other type, you have to invasively get the cells, whereas skin, I can go like that and I just have a bunch of skin cells now. So it's pretty easy.
Okay.
Does anyone have any questions?
Also, does it have to be, like if I'm putting it, does it have to specifically go back into my body, the ICS cells, or can they be programmed to go into somebody else's body?
That I'm not 100% sure on. In my opinion, it would probably be best if we're doing that like for your cells to be used because it has your DNA, it's your genetic code. But remember, there was the case with the mice where they took the cells from them, but they were still rejected.
Has there ever been a case of that like humans where they've been rejected like that, or is it?
Honestly couldn't say. If you Google it, it'll probably come up, but I couldn't say off of what I know. I don't know if you guys.
So the idea with cancer, one of the theories that's kind of going around is that stem cells might be part of what is causing cancer. So basically, what's kind of being hypothesized or tested is that there, like Carl was saying, there are more mutations in the cells, and so that causes problems inside of them. That they're not functioning as well as they should be. And then if the cell has some kind of problem with its division regulation, it kind of just goes out of hand and it just keeps going. And there's no way to stop it from, let's just say, like invading the whole body or creating a tumor. So that's one of the ideas is that the stem cells or the cancer cells are just unregulated and they keep going, keep dividing.
Does anyone have any questions on stem cells or cells in general? We're going to be switching gears over to signal transduction, so if you have them, any last comments, thoughts, questions? Yep.
So the theory is that stem cells can cause cancer?
The theory is that these stem cells become cancerous. So we'll talk about starting on Wednesday. There's a lot of checkpoints. So do you guys remember cell division from your last bio course that you probably took? There's a lot of checkpoints to kind of prevent you from going into division if it's not supposed to happen. So in cancer, what's happening is these checkpoints are blocked, and essentially there's nothing stopping it from just dividing. And so because we're talking about division, and then you get all these uncontrollable cancer cells, that would be in stem cells because those are the cells that are dividing.
The picture underneath that, though, is that saying that it can also like tear itself or something?
Right. So if you can get the cancer stem cells, which are the CSCs, and you can knock those out, you can if you kind of figure out what's going on, you can shrink the tumor and get rid of it. So if you target for those stem cells, it's fine. Whereas the other ones, which is the standard like chemotherapy, it's a clear cut of all those cells. So if you target it to be more specific, you wouldn't lose the cells that you're losing with standard therapy, and you'd have more success getting rid of tumors.
This has to do with ICS cells. So say when I'm putting it back into my body, can my genes are they still being expressed when they're developing into my new liver cells?
Yeah, so there will always be genes expressed. So what happens is there are 4 genes. I don't remember all the names, like SOX and that's actually the only one I remember. But those 4 genes are the differences between pluripotent stem cells and the cells that they used. So what they essentially did was they went back and reconfigured what was being expressed to look just like the stem cells, thus making it basically be a stem cell. And then it can go through the process of dividing to make more stem cells or differentiating.
Okay, so the genes are still being expressed. What about the DNA? Is that going to stay the same?
The DNA, so across all your cells, the DNA is the same. So when you're going through these process and you get genes expressed, that's transcription translation. You guys might have had that in previous courses. We're going to be talking a lot about it coming up. But essentially what's happening is your DNA is copied to RNA, and then that becomes protein. So you're never making changes to your DNA, you're just kind of copying it. to then change it. So your DNA across all cells, like we said, is going to stay the same. Unless you accrue mutations and that changes the DNA and it gets broken. So that can change, so it's not an always bad thing, but typically it's the same. Does that make sense? Any other stuff?
Okay. Basically, we can have cells in our body that are not receiving signals. So even like survival signals.
Sorry for the grammar. I totally didn't know what I was doing.
Survival signals are always there. So if a cell does not receive any signal, it goes apoptosis, which is programmed cell death. So just keep that in mind that it's always receiving signals if it's in the body. Even stem cells that are not differentiating, even if they're not doing anything, they always have cells to keep them alive, signals to keep them alive. Different cells would react differently to each signal. So, for example, like a gland would produce something, but a muscle would like contract, and cells would react differently to even to the same signal. Like it depends on its function, where it is in the body, and what it has to do. Okay. Steps of signal transduction. First of all, it has to receive a signal. So right there in the signal. And then the receptors dimerize, which means that they come together and they form like a complex with the signal. And then one of the receptors, they cross phosphates, so they activate each other. And TK1, when they phosphate each other, TK1 comes and is phosphorylated, and then it activates TK2, and then it activates TFA. TFA is not a kinase; it's a transcription factor, which means that it goes into the nucleus and it marks part of the gene where it should be transcribed. So it just makes it more stable for the RNA to come to the gene and transcribe.
So something with this diagram that you guys should pay attention to is that it's kind of like a step-by-step process. So when you get questions and when we've done clickers, you can think, what is step one? Signals received. Think of it kind of like a flow of events. Signals received, dimerization, go through the steps, write them out if that helps you. And when you're answering these kinds of questions, refer back to it. So on the test, actually, write that down if that's something that helps you. So keep in mind, think flowchart kind of thing, similar to this.
Another thing to remember is that they're not using the same phosphate. So each, like TK1 is phosphorylated using another phosphate than TK2. They're not like passing it down. Each one has a new phosphate.
Yep.
Where does the phosphate come from?
The phosphate comes from ATP.
Adenosine triphosphate. So they take a phosphate is removed and it's put on the TK1 or whatever that's being phosphorylated, and then you just have ADP, which is adenosine diphosphate.
And that's what the job of a kinase is to basically take the phosphate off of ATP and put it on the next one. So these receptors, they help each other get their phosphates. Then they help TK1 get its phosphate. TK1 helps TK2, which then helps TFA, and so on. So they're almost like helping each other. But just keep in mind that they're not passing that phosphate. That's really important. Yes.
So where is ATP in this?
It's not shown here. So energy is just, ATP is energy, and it's just in the cell. So the mitochondria in the cell, if you know your organelles, that's what produces energy. And so that's just in the cell. It's not really being shown here. Because you guys don't need to worry about how it actually takes the phosphate off and puts it on.
But in order for it to phosphate, ATP has to be there?
Yes. And you can kind of see that here.
And it's
So what?
there.
Yeah, so basically ATP is always in the system, and as she said today, if ATP is not there, the cell dies. Because it's out of energy. So what's happening is, in this case, there's no phosphate group. This is, we'll look at it like, let's pretend it's TK2. ATP comes in, it undergoes hydrolysis of ATP, which you don't need to worry about that word, and then it has a phosphate group added. The kinase does this. It puts the phosphate group on. The other side of it is when you remove the phosphate. And essentially, the phosphatase is the enzyme that removes the phosphate group, and that takes it off and affects this. And we'll talk about whether that means activation, deactivation, or what as we keep going. Any questions?
Yeah, I just wanted to go back to the signaling. So the same signal can be, I don't get that. Yeah.
So basically, in this case, so in this case, we're looking at acetylcholine, which is a neurotransmitter. And what we're saying is depending on where it binds into what tissue, what cell, it's going to have different effects. So in heart muscle, when acetylcholine binds receptor, the cascade happens and you get decreased rate of contraction in your heart. I think that's part of the flight or fight response. Salivary, the receptor binds and you get secretion, which means you get production of saliva. So would you expect your heart muscle to start salivating when it binds? No. So that's the thing, is these signals, instead of having thousands and thousands and thousands of signals for all the different things, the cell can interpret based on how that signal binds and what that cell does, how to use that. You see it again here when you have acetylcholine bind to the skeletal muscle, it leads to contraction. Does that make sense?
I don't know if you're getting to this or something. I just have a question about like kinases. So in that diagram, the cell signaling thing, where do kinases and like cascades come in that? Like
In
this cell.
in this one?
Yeah.
So these are the kinases. So TK2 just means tyrosine kinase 2. So the R stands for receptor. The TK is tyrosine kinase 1, 2, just because, I mean, it doesn't, there's no 1 or 2 in these cells, it's just for hypothetical. And TFA is transcription factor A. There could be transcription factor B, C, X, Y. It's just a name.
So, did you see like a ton of questions referring to like the disruption of the transduction?
So like, you will see questions on the test, at least I can assume, I haven't seen the test yet, that you have this flow. Great, you understand it. What happens if we remove the phosphate from TK2? What happens if we completely remove TFA? So that's why I suggest definitely drawing that flow out, because if you have the flow of the events, you can say, okay, we have TK2 removed. Everything before that is going to happen normally. Everything after is going to be a problem. So you can kind of use that to visually see what's going to happen, and then you can go from there based on what you know.
I remember like it was a couple of questions. It wasn't just that like simple. Like it was always like that a little trick to it. Like even if I do the diagram, but like wasn't there a question where like it was like TK2 is gone or something? Does it connect? And it was like, but there's many other. Like,
So that's the thing is, if it's a multiple choice, it's going to be there's one straight answer. If that was an essay, so or a short answer. Let's pretend TK2 is removed. In this pathway that we know, can you get transcription of the gene? No. But what could happen is the activation from another pathway could lead to activation of TFA. So essentially, if you were writing the short answer, you would want to say, in the pathway that we've been using, you would not see TFA be activated unless there was another tyrosine kinase that could activate it, unless there was another pathway that could interfere with it. If there was, so this is why we get the short answer question is designed to tie all the different pieces that you learned together into one. So it's kind of like the critical thinking part of it. You've been hearing that for a while.
Can you explain what TFA does again?
So what TFA does is it's a transcription factor. So transcription factors bind to the DNA and lead to differential gene expression, which basically means that by binding transcription factors, different genes will be produced. So in this case, if we're looking at the salivation example from the previous one, the transcription factor leads to production of genes that lead to salivation. So it's basically going to cause an outcome and you're going to get the genes needed for that outcome. Does that make sense?
Did she say we're going to get this diagram on the test?
So any pictures and diagrams like this, the ones that we were working with today, you guys will get on the exam. So she doesn't expect you to memorize these things, she just expects you, I mean, we could give you this, and if you can't explain it, it doesn't help. So you have to be able to take these pictures and understand what it's saying.
I just wanted to hear that one last time. If what you said about the short answer. So if TK2 is off, then that pathway doesn't work, but the TFA can still be transcribed into the nucleus through another pathway.
So if there was a receptor here for something completely different, and one of the effects was it interacted with this pathway, it could, in theory, work on that. So another example, if TK1 is the thing this time, if you have another pathway that can activate TK2, it'll finish. So basically your goal is, again with the flowchart, think of what's being disrupted and what you can do to fix that disruption.
And just to be clear, I don't know, I think you said TFA gets transcribed. Make sure you keep the terms separate from one another. So TFA is activated, it goes into the nucleus, and it leads to transcription of DNA. So TFA doesn't get transcribed. So you just want to make sure
The nucleus gets transcribed, right?
No, the DNA inside of the nucleus will get transcribed.
So remember that
it's
when
inside the nucleus, so that's good to keep in mind as well, the location.
So keep in mind that this whole thing is regulated. So you'll notice TFA in both cases is outside the nucleus, away from the DNA. When it's activated, it then can translocate, which means go into, and work on the DNA to get transcription of the DNA to get the genes that you are essentially turning on. So like Samira was saying, you want to keep your terms right because you do get partial credit on these short answers, but if you said, for example, the TFA is transcribed, that'd be wrong. So you'd get points off for that. So just be careful with your terms. Don't oversimplify things, but don't go too complex because then you might wrap yourself around and have problems. Do you still have a question?
So when we say like these questions, like if that was disrupted or something in the pathway or like turned off, like how does it get disrupted? Is that like a mutation or something?
It kind of depends on what's happening. We could mutate it and say we make the mutation in TK2 so it cannot be phosphorylated. That works. But if we have, which we'll talk about later, ubiquitin-mediated proteolysis, if we undergo that and take that away, that's not really a mutation. That's just what process happening. And then
we don't know why these turn on.
Right. So you have to kind of, in these short answers, make an explanation based on what we talked about that makes sense and can be explained. So you're basically playing house, as in Dr. House, the TV show, not, you know what I mean. Yep.
So does transcribe mean like different genes in the
No, so I'll draw it for you guys. So did we talk about the central dogma? Yeah, a little bit. So basically, all life is DNA to RNA. That's going really loud. Protein. And so what's happening through this is you get DNA to RNA is transcription, RNA to protein is translation. So the reason they're called that is because you're taking your DNA code and you're transcribing it, transcription, into RNA code, which can be read by cellular machinery to then translate the code into actual protein. So you're transcribing, basically your DNA is the recipe book. You're transcribing your recipe onto your recipe card, and then you're translating that into making the pie that you're making. Does that make sense? So when we talk about that, that's different from differentiation. Transcription, translation, the end result is protein.
What's the RNAP? Like, where does that come in before the DNA?
That is RNA polymerase. So that is the thing that takes DNA and transcribes it into mRNA. So this binds with transcription factor, so they can all they form a complex that that basically targets a certain gene and that leads to the transcription.
Are there, oh, I'm sorry. There's a difference between transcription, translation, and just being transcribed.
Yes. So when we say transcribed here, we're talking about the whole process, transcription, translation. We haven't gotten that far yet. So in about a week and a half, we'll start with this, and we're going to get into every detail about transcription, translation that you never wanted to know. But for now, just to keep it simple, we just say transcribe because we're not showing the protein being made. But with this means transcription, translation. The end result is a protein. Yep.
So not all cells express genes, right? But when wouldn't they? Like when don't they express?
All cells do.
Oh, also.
All cells do.
Okay.
Unless they're apoptosing, which even then to apoptose they have to express a gene.
About the central dogma theory, I don't know if Laura ever mentioned this, but she might have. So the central dogma theory, this DNA, RNA, protein, that is an accurate, series of steps, but it's not, don't take it as 100% correct because I don't know if she wants you to know this or not, so I'm sorry if she doesn't, but RNA to DNA, the flow from RNA to DNA does exist in viruses. So there have been debates and conflicts over whether or not the central dogma theory can flow backwards, and just know that it can.
So if you Google the central dogma, you'll see either this flow or you'll see the same flow with all these arrows kind of going around because viruses really screw things up. Because they can take their RNA, which is their genetic information. So that kind of goes back to that first discussion question that you guys had. They take their RNA, put it into the host, and basically it's called reverse transcriptase. You don't need to do that anymore with that, but it goes in reverse. And they put their genome into the DNA of that cell. So then if their genes are now in the DNA and you go through this process, you're spitting out viral protein instead of your own protein. And that's how they hijack a cell. And then they start and they get all these cells, and then that's how you get viral infections, essentially. Don't worry about that too much. Just kind of keep in mind in terms of that discussion question.
Just know that the backward flow can happen.
It's
That's all you need to know.
One of the quick questions is the signal transduction, change gene expression, was false, not always.
Which quiz was this?
I think it was the two.
Does she have an explanation on that?
No.
She might be getting at the fact that it might not end up activating the TSA and then it doesn't go into. I don't know if that was her line of reasoning, but
Oh,
it
yeah.
might be that.
That's a good one. Was it, you said quiz 2?
Yeah.
Was that the online version or was it the practice?
The
I don't know if I can actually get into that. So remind me about that and we can hit that at the end. So basically, do you want to just repeat that?
Yeah. So signal transduction is basically when you have the signal going from one kinase to another to the TFA. However, this question might be getting at the fact that the transcription factor never ends up, or in some pathways that are mutated, you might not have the transcription factor getting activated. So even though you do have signal transduction occurring, you don't have the last step taking place. So you can't have the gene getting expressed.
Okay. So we talked about the phosphorylation and how you put the phosphate group on, but what effect can that have? And so a lot of people kind of just assume phosphorylation means activation. In everything we've talked about so far, that's pretty much been the case. But don't hold on to that. So this picture is a really good representation because it shows kinases are kind of like an on or off switch. In the top one, the kinase puts a phosphate on, and that activates this protein. The phosphatase removes it, and it turns it off. That's what we've seen with TK1, TK2, TFA. The other side of things is when it's not phosphorylated, it's on. But when you have that kinase put a phosphate group on, it turns it off. So the act of putting a phosphate group on leads to a change in conformation, which is shape. It's really important, if there's one thing you learn while you're in this class, it's that structure fits function. What that means is a protein's structure is fit for its function. Think about it in terms of like a cheetah. A cheetah is designed physiologically so it can run as fast as it can. If you tried to run as fast as a cheetah, it wouldn't work because your structure is not made that way. So it's the same thing with proteins. They are shaped a certain way to do their job the best way they can. So when you add these phosphate groups, you're changing their shape. And then you can see that because now there's a little lip on both of those. Depending on the shape that's best for function, adding or removing that can lead to different effects. So hold on to that. If she gives you an example on the test where she tells you putting a phosphate group on turns it off, don't get surprised because that can happen. Also, don't assume that when she's talking about in the short answer that putting a phosphate group on always means turning on. So if you're given a hypothetical pathway that's a little different, and she'll give you the pathway and say, work through this, you have to keep in mind that both could happen unless she says otherwise. So hold on to this. This is one of the more important pictures to hold on to because a lot of people mess that up.
So are you saying that that kinase can actually turn off another kinase?
By putting a phosphate group on, it could lead to turning off.
Oh, okay.
So in the case that we look at, TK1 adding a phosphate to TK2 activates it.
Right.
But there are pathways where that could deactivate it.
Oh.
How do you know when putting the phosphate on that it turns off?
You have to do the experiment essentially. So basically, we're not looking for you in the short answers to prove, yes, this is what's happening, but you have to, unless you can rule it out, it's a possibility. So if you were given, we have this new pathway, does phosphorylating TK, 73, keep the pathway going or not working? In that case, it would be, well, if it is turned on by a phosphate, it would keep going. If it's turned off, it can't. You probably won't see something that direct, but just kind of keep in mind, it'll probably pop up in some form on the test.
A lot of times on the exams, she tells you if it's gonna activate or deactivate. It depends on the question, but if that's like needed information, she's good about giving that to you.
And she'll give you everything that you need. So sometimes you'll read the question and you'll be like, crap, what is she saying? I don't know how to do this. This is a new pathway. That's not fair. But she'll tell you everything that's different from what you know. So essentially, if she gives you a new pathway that has an extra couple kinases, she'll give you everything you need to know about that so you can answer the question based off of what you know. So if it's something that looks a little strange, ask one of the TAs and we can kind of help you understand what's going on, but she's going to give you everything that you need to answer the question. So I know that some of her stuff is really wordy, but just try and break it through and pull out what's important. And again, ask us for help. Feedback loops. Maybe you guys love feedback loops.
So a feedback loop is essentially when you have, you get out something from later on in a chain that goes back and either upregulates or downregulates the rest of the pathway. So for example, if you have transcription, or if you have TK1 activating TK2, which activates, let's say, the transcription factor causes the gene to produce TK3, just hypothetically. So let's say TK3 comes out of the transcription of the gene and it goes back and it activates TK1. So that means that it is upregulating or it's in a positive feedback loop. So by expressing the gene, you're causing TK3 to come out and make more expression of the gene. On the other hand, let's say TK3 were to go and deactivate TK1, that's going to be downregulating. That's a negative feedback loop where expressing the gene basically causes the pathway to shut down. And then there's the whole, I don't know if you guys remember, she drew the oscillations on the board the other day. That's basically with negative feedback loop. So basically an oscillation is showing that you have, so like let's go with the TK3 example. So TK3 is going to go back, it's going to deactivate TK1, which is going to shut down the entire pathway. By shutting down the pathway, you end up having less or no TK3 in the system. By reducing the amount of TK3 in the system, you're turning the pathway back on because there's nothing turning the pathway off. So the system turns back on, TK3 gets produced again, it goes back, it downregulates, and it kind of cycles. So that's what that oscillation is. And that's a kind of confusing concept, so if you want us to explain it again, just let us know.
Could TK3 ever like destroy TK1s or like?
So it could, in theory, you go through this process, you make TK3, and that in turn activates a phosphatase, which then goes back and turns TK2 off. You could also have it activate ubiquitin-mediated proteolysis, and that removes that. You could have it do receptor-mediated endocytosis. So it can have different effects, and that's something that's actually a really good point. Is if you're given a question on the exam that's like a short answer question, those are all valid things that you could say, like, well, what happens if TK3 does this? Different outcomes.
So does that go on forever? I mean,
Essentially, yeah. So basically, you get production of the gene, and you get production, but TK3 in turn stops production because it's inhibiting the pathway at TK1. So production goes down because now you're stopping. But because you're stopping it, you're no longer making TK3. So there's nothing stopping the pathway, so it starts back up. But then you're making what's stopping the pathway, so it stops again. And just kind of keeps going. And this is all under the assumption that signal is still bound.
So yeah, so as long as that signal doesn't change or anything.
As long as signal is bound, it'll keep going. If signal leaves, it'll stop. And basically, what'll happen is you'll get production, it'll stop the pathway, and then if signal's gone here, it's not going to climb, it's just going to be it's done.
So do TK3s like end up like disappearing?
Essentially, they're like being used and then they're gone in this
But
case.
they're temporary. So like say like TK3s are always there, then that essentially like if TK3 stops TK1, but they're not temporary. Say they're forever, don't they just like stop TK1s forever?
Essentially, they equal each other out. So if signal's always bound and TK3 is always inhibiting, unless one has more force and is stronger, it's going to be like an even match. You're not going to get production, but at the same time you have it inhibit. So it's kind of like, Alright, you can skip back to that.
Ubiquitin-mediated proteolysis. Basically, when we finish, like proteins are produced, and they're used up, and then we need to they need to be thrown away, degraded. So proteasomes do this degradation. Basically, ubiquitin ligase, which is like an enzyme, it labels the protein that needs to be degraded, so it just marks it, and then the proteasome basically comes and it degrades it.
So essentially what's happening is, if you didn't have proteins degraded, they would just pile up and pile up and pile up, right? So if you don't throw your garbage away, it's just going to pile up until it's, it's everywhere. So we break down proteins when they're no longer needed. What's happening in this process is essentially you have these ubiquitin tags, and you don't need to worry about really what they are, but they're basically tags that say this is okay to be thrown out. So essentially, it's kind of like you taking your garbage and putting it in the trash barrel, like outside to go, you're basically putting a tag on it that says, this is okay to be deleted, get rid of it. And so anything with that tag will be put through the proteasome and will be broken down.
How does it get tagged?
Through this process, it'll get tagged. I don't think you need to know like the logistics, the step-by-step of how it works.
It's just that the ubiquitin ligase like kind of sticks on it, so it's like marks it. That's it, basically.
Is the ubiquitin ligase like similar to the proteasome, or like what's the difference between the?
That's just the tagger, and then the proteasome is the thing that actually like cuts it up. So it's essentially two pieces to this machinery. So something tags it and then something removes it. So the other maintenance kind of thing is receptor-mediated endocytosis, and we talked about this today. And so essentially what's happening is you remove the receptor from the surface. So when the receptor is no longer needed, it can be taken in and degraded, and then in this way the signal can no longer bind. You can also take it in, as we talked about, and to remove the signal. So essentially you're taking the receptor off this surface, so the pathway cannot be turned on again. The receptors can then, if once you remove the signal, they can go back out there, but they are pulled off. If they're degraded, then they go through degradation, they're gone. I don't know if it's the same thing as ubiquitin-mediated proteolysis, but they go through a similar process.
I thought Homer said something about like during this process, there's only some receptors that are being recycled while there's also other ones that are still on the outside.
Yeah, definitely. So this is not for every receptor. This is again a regulated process that can happen. So this is just like not all proteins are degraded at once. It just kind of happens, and the cell knows how to time it for that. What you guys want to make sure you know is that, for example, the first clicker today, and it said, are the receptors present? And it said yes. So you could rule out receptor mediated endocytosis. So you want to know what it is and that it is possible. So if the receptor is missing, this is probably what did it.
Remember that cells are constantly receiving signals, so you don't want all the receptors to be eaten up, because then the cell is going to die. You always need to be at least receiving she calls them survival signals. So that's basically like the food, food and like growth.
The not die. There's literally one that's like do not die. Whereas if you think about going back to stem cells, they have signals for GF, which is growth factor. When you want a cell to divide, you need it to grow so it can become two cells, right? Would you want a differentiated cell to be receiving those signals with a receptor? No. So that would be a good example of it goes through differentiation and then all those receptors are endocytosed and recycled. So we talked about the backside of your packet, of the piece that you got today, and based on the clickers, there was some confusion, so we will go through this to kind of make sure that everyone's on the same page. If you look at your handout, it gives you these pictures in a smaller view, and it gives you paragraphs below that explain kind of what's going on. She usually leaves that stuff on there, so those are your explanations. You can use that if it's provided on the exam, but make sure you understand what's happening. So that's what we're going to do. We're going to kind of walk through it. So this first one is the first picture that you see. This is the quiescent stem cell. This basically it's just kind of there. It's not dividing. It's not differentiating, and it hasn't received the self-renewal signal. Is it receiving signals? Yes. So it's receiving the do not die signals. It's receiving all of those survival cues that we're talking about. So just because it's quiescent does not mean it's not receiving signals. And so what's happening here is there's a little legend of what's going on. SR is the self-renewal signal. DET is the determination signal. IB is inhibition of differentiation genes. DIB is the genes for cell division. Skin is differentiation skin cell genes, and neuron is the neuron genes.
What is self-renewal? What's self-renewal?
Self-renewal is the first thing that you have to do. So it leads to either differentiation or division. So basically what's happening here is before the signal is received, self-renewal puts these pieces of machinery in place. Because before it can really do anything, it has to have your TK1, TK2, TFA, all that stuff. Yep.
So it has signals, but it's not signals through the process?
No, so basically what's happening is in all quiescent stem cells, they're not receiving signals yet, but the genes, the SR genes for detecting self-renewal, they can't really detect signal unless this leads to these.
But they are still receiving.
Yeah, oh yeah,
Yeah, they are still receiving
like the survival
basic signal, but they're not receiving anything else for like to lead them to differentiating or dividing.
Okay, just survival.
Sorry,
Yeah, just survival.
And they talk.
They're being told not to go into
Okay.
apoptosis. Apoptosis is the programmed cell death, so if they get that signal, they're just done.
So have they like not received the self-renewal signals like by random, or is it like for a reason that they aren't self-renewing yet?
So these are the stem cells, so they have, before this gets activated, there's no receptor, there's no TK1, 2, or TFA, so it literally can't do anything with that pathway. So what happens is before it starts dividing and differentiating, this produces the components to then make it available to receive signals, essentially. So this is the first step, essentially, is putting the things in the pathway. Does that make sense? So it's not really receiving signals, and this doesn't just kind of happen. It's in all stem cells, this happened, and all this stuff is in the pathway.
Okay.
So it's not being activated, it's just kind of like, here we go, we're putting everything in place.
So in this one it says there's no signal for self-renewal. Like what happens at that?
So there's nothing telling it to self-renew at this point. So it's not one of the survival signals, it's just,
So is that ever gonna self-renew?
it will when it receives the signal. So in this snapshot of time, it's not receiving the signal yet. When it does receive the signal, which is actually the next one. Yep.
So quiescent stem cell, it's not dividing, not differentiating. So say it's just a regular skin cell, but it still receives signals that tell it not to divide.
So these are stem cells, so they're not differentiated yet. They're just basically sitting there receiving only the signals needed to survive. They're not really doing anything else.
Would have went through all of this process and then they became thin, and that was it.
So what makes it a quiescent stem cell?
It's basically any stem cell that has yet to go through self-renewal. So it's basically when you think of, it's like kind of like which came first, the chicken and the egg. If you have a cell that's not dividing and not differentiating, it's probably in quiescence.
And this is the version when it starts to
Exactly.
Self-renew.
Exactly. So now you can see the self-renewal signal is present, binds receptor. This is the same pathway that we looked at before, TK1 to TFA. TFA affects three things. It affects division, so it's preparing to divide. It's affecting the ID, which is the inhibition of differentiation. So you don't want to undergo differentiation while you're dividing. That's really bad. So what it does is it activates this, which stops differentiation. The third thing it does is it sets up for determination. And what that is, it's kind of like the SR, what we looked at before. It activates these genes, which puts these components in the pathway. So they could receive those signals. So again, division, inhibition of differentiation, and determination. Good? Yep.
So once you get the self-renewal signal, then the determination signal can happen, and it can then make receptors that would accept other signals to differentiate into different types of.
Right. But until this division and this step is done, so for this picture, if it were hypothetically to receive a neuron signal and it went through this whole process, you're inhibiting, thank you, differentiation, so it can't undergo that because, like I said before, you don't want to be dividing and then be like, oh, let's become a skin cell at the same time. That's just bad.
So it's right before that process.
Right. So at this point, you're activating the genes for division. And so you're going to divide in this case. Okay. So for the next one, this is after you've gone through the division, stuff like that. You're no longer expressing division genes because you've divided. And now you've received your skin signal. Because you've gone through division, you're no longer activating inhibition of differentiation because you're ready to differentiate. So what happens is, signal is received, TK1, TK2, TFA, same bit as before, and it comes down and does two things. Actually, three things. First is it inhibits the inhibition of differentiation. So you're inhibiting an inhibitor, which basically means you're stopping the thing that stops you from doing what you want to do. Does that make sense? Okay. At the same time, it's activating differentiation of the skin pathway, which is good because you're receiving the skin signal. When this gets activated, it does two things. It activates upon itself to undergo differentiation, but it also acts as an inhibition for the differentiation of neuron pathway because you don't want that pathway coming on. You also see a similar thing happen up here where it somehow blocks. So you're basically stopping in two places the neuron pathway from happening. Because if you're in the middle of differentiating into a skin cell and then you get that neuron signal, you're going to get a sneuron, which is really bad. It's not going to do its job. One little thing for reading these, if you can kind of see how that's an arrow with a point, that's an arrow with a point, that means activating. When you see the line with a T at the end, it means inhibition. So this is inhibiting this. This is activating this. Good? Yeah.
So you said before that the TFA does three things, but yet it changes what it activates for what for each pathway. So for
You repeat that? Sorry.
differentiating pathway, TFA does different things than.
Well, these are different. So this, I'm just using TFA in general. This is one transcription factor. This is another transcription factor. This is another one. So if we wanted for naming sake, this is TFA, this is TFB, that's TFC. So they're different ones. I'm just trying to relate it back to that one pathway, the hypothetical. So different transcription factors.
Can you explain the previous slide again?
Yep. So what's happening here is self-renewal. So you get your self-renewal signal, you go through the cascade that we talked about before, and you get three things. Activation of determination. So you're activating these genes, which in turn makes your TK1, 2 in TFB, your TK1, 2 in TFC. So you're making the pieces for this, for both pathways. You're also activating the ID, which is inhibition of differentiation, sorry. So basically what that does is it stops you and inhibits you from undergoing differentiation of either type because you're undergoing division. With that in mind, you're also activating the division gene because you want to divide. Does that make sense? Yeah.
The cascade what goes through right before it goes into the neuron? What is the cascade?
So the cascade is this. That's the signal transduction cascade. A cascade is essentially a series of events that happen. So when I say cascade, I mean this entire event is the same here, here, and here. There's just different outcomes. So essentially we're looking at the same hypothetical 3 times, but now we're going a little bit deeper because we're looking at what it does, and they're having different effects. Does that make sense? And so the neuron is pretty much the same as the skin, but just, for clarity, we'll go through it. Neuron is received, this cascade of events, this pathway that we've been talking about so much, it goes through, you get TSC in this case, leads to the same thing as the skin except for neurons. So what that means is you're inhibiting the inhibition of differentiation. You want differentiation to happen, so you stop the thing that's going to stop it. You're activating differentiation of the neuron because you're in the neuron pathway, which then activates itself because you really want it, but it also stops the skin differentiation because you don't want to start becoming a skin cell in the middle of becoming a neuron. So those two are pretty much the same. It's just if it's a skin, this is what's going to happen. If it's a neuron, the opposite is going to happen. Do these four pictures make sense? Okay. So we went through these clickers and they didn't really go 100%, so we're just going to run through them. So a researcher tests for just 3 genes, just 3 of the genes in a stem cell, and finds the following. So the gene that encodes SR is transcribed. So what I'm going to do, write that here. We've got skin TK2 is also transcribed, and ID is transcribed. So if we go back to our pictures, we have SR being transcribed. We have skin TK2 being transcribed, so it's being put in the pathway. And we have ID being transcribed. So what's happening, SR, ID is being transcribed, and determination TK2 is being transcribed. So in this case, it's the SR signal. Does that make sense? So these are the kinds of questions that you will probably see on the exam where she's going to give you what's present or not, or these 3 things are happening. Stuff like that.
So it's like the skin in the neural pair underneath the
Right, so you're undergoing this pathway, and then the determination is on, which puts those components in. So there's no signal bound to skin or neuron. So notice how the arrows are moving towards the receptor and not towards the nucleus. That's because essentially it's making that and putting it in place. It's making this and putting it in place. Yeah.
So the skin pathway and the neuron pathway can happen unless the SL pathway happens first.
Right, because otherwise those components just aren't there. There's nothing to replace.
Yeah, so what's the next step if it was going to become a skin pathway or go down the neuron pathway?
What's the next step from here?
Yeah, how would it look like since you wrote one is ID, one is inhibit differentiation gene, so would it just be like?
So in that case, it would look like this. You bind signal, and it comes through, blocks this, turns that on. And then the same thing for the other one, except it's going to target this one. And so the answer for that one was SR. Really crappy image. Okay, so for this one was a little bit tougher. We didn't get to it, so we'll go through it now. When skin cells are fully differentiated, they no longer divide. Right? You know that. So, which of the following could cause the division gene to stop expression? So we're looking essentially to stop division. So the differentiation for skin genes produce proteins that activate receptor-mediated endocytosis of the SR receptor. Does removing the SR receptor stop division?
Yes.
Yes. So if we go back, if this is gone, do we get division?
No.
Right. So that's a yes for what was that thing. The same if the differentiation skin genes produce inhibitory transcription factors that bind to the SR genes and prevent expression, will that work? So what that's saying, I know that's like a mouthful, it's saying you're getting production of transcription factors that prevent this from doing its job. So would that affect division? Yes. Because remember, in the first one, you jump back one. If this is not working, or is any of that going to happen? No. Okay. The next one was the skin genes produce proteins that activate ubiquitin-mediated proteolysis of the phosphorylated form of the SR pathway transcription factor. So visualizing the pathway, if we target the phosphorylated transcription factor, are we going to get division? No. So we'll get it. Sorry, someone's jumping back and forth. So if we take this out, are we going to get division? No. And then the last one, the differentiation skin, again, genes produce a phosphatase that targets TK1 of the SR pathway. Will that pathway run if we remove the phosphate?
No, it wouldn't.
Right. The only exception, which is not for this case, but what if removing the phosphate were to activate it? Would that be the pathway would run? Yeah. So in this case, we wouldn't, because that's not what happens, but just in this way. So all of those are the right answer. So for in the lecture notes for today, both of those questions are explained as well. So feel free to check those out. That's it. So if you guys have any questions.
Is the test gonna have like 4 answers?
No. So the